Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to all to Bioscience with Shatin Sawati. And I'm here to deliver the lecture from DNA sequencing. And today I'm going to discuss about the Maxam-Gilbert sequencing. I have discussed in detail from the biotechnology series about the basic principle of the sequencing and the Sanger method and cloning. So must watch all these lectures. So this lecture will be easily understandable for you people. So Maxam-Gilbert method, I got a lot of questions about the Maxam-Gilbert method because students are so much confused about the chemical cleavage and how the fragments are produced and many more. Maxam-Gilbert method is little bit technically complex than the Sanger method. That was the reason that it was fallen out from the DNA sequencing. So I will discuss each and everything in detail. So keep watching all my lectures. Do subscribe my channel, like my videos and share my videos. Okay, now I am going to discuss and give you the little bit information about the history of the Maxam-Gilbert method. Why it is called Maxam-Gilbert method? Because it was discovered by the Maxam and the Gilbert. So that's why in honor the method was known as the Maxam-Gilbert method. This method is based on the nucleobase specific partial chemical modification. This method based on the chemical modification of the DNA nucleotide and DNA bases, nitrogenous bases, nucleotide bases. So it depends upon the chemical modification, and the second one, the subsequent cleavage of the DNA backbone at site adjacent to the modified nucleotide. First one, what was the first one step? The chemical modification, and the second one is the cleavage of the DNA backbone at the site adjacent to the chemical, where the DNA is modified chemically. There is a break or cleavage of that at the DNA backbone. Is it clear? So I will show you how. On the right side is the Maxam, and on the left side you can see the Gilbert. I'm paying tribute to the great leaders, legends, and actual the leaders and honorable person and the real hero. Okay. Alan Maxam and Walter Gilbert in 1977 published a paper. A new method for sequencing DNA was honored by a citation for the Chemical Breakthrough Award from the Division of the History of the Chemistry of the American Chemical Society for 2017. It was presented to the Department of the Molecular and Cellular Biology, Howard University. Is it clear? Okay. Now, what was the drawback? Must keep it in your mind, the Sanger method and the Maxam-Gilbert method both were considered as the first generation of DNA sequencing method. Now, I will discuss in detail about the next generation sequencing in my next lecture, so must watch all my lectures one by one. So, Maxam-Gilbert method was you can say fallen out of the favor due to its technical complexity. It was technically complex, and you will see, and prohibiting this technical complexity. What will happen? Prohibiting its use in the standard molecular biology kits. And the most important thing is the use of the hazardous chemical and difficulties with scale up. That is very much. That's why it was fallen out of the favor. That was the drawback of the Maxam-Gilbert method. Now I'm going to discuss the steps of the Maxam-Gilbert method. That is very easy. The basic principle is same. The collection of the DNA, first of all, means the preparation of the sample. And how we will prepare the sample? First of all, we collect the DNA of any species, any organism which is unknown, and then first the denaturation of the double stranded into single stranded. As you can see here, the double stranded is denatured into single stranded. So I will not discuss in detail how and why, because I have discussed in my previous section in detail. Now the next one is the most important, radioactive labeling. How the radioactive labeling was done by the Maxam-Gilbert. At the 5 end of the, you can see here, this is the single stranded DNA, and at the 5 end of this single stranded DNA, the radioactive isotope was added, and that radioactive radioactive isotope is phosphate, means with P32, phosphorus P32 was added at the 5 end by the Maxam-Gilbert by the kinase reactions, using the kinases enzymes. Is it clear? So first one is the preparation, then denaturation, and then the second step is the radioactive labeling, and labeling will be done with the P32 phosphate, which is the radioactive isotope at the 5 end. Is it clear? Now the chemical treatment, that is very important and little bit complex, but it is very interesting, so must listen carefully to the chemical treatment. You can easily understand the basic concept. Now the chemical treatment generated breaks a small proportion of 1 or 2 of the 4 nucleotide bases in each of the 4 reactions. So Maxam-Gilbert method carried out the 4 reactions. One reaction, you can see here the 4 tubes, and these tubes contain, first tube contain the G nucleotide, second one with the G plus A, mean the guanine and adenine, the third one is the thymine and cytosine bases, and the fourth one with the only cytosine base. Is it clear? Or the cytosine nucleotide. So you can see 4 reactions were prepared by the Maxam and Gilbert. So first of all, what will happen? The chemical modification or the chemical treatment with the purine was done with the formic acid, and purines, you are familiar purines with purines adenine and guanine. So adenine and guanine are depurinated using the formic acid. Modification was done with formic acid and the dimethyl sulfate. Must keep it in your mind. The purine, adenine and guanine are treated with formic acid and dimethyl sulfate. And the pyrimidine, which are cytosine and thiamine, are hydrolyzed using the hydrazine. First, purines are chemically treated with formic acid. Must keep it in your mind and note down. Okay, note down on your notebook. Formic acid and dimethyl sulfate is used for the purine, and the hydrazine was used for what? Pyrimidine, means cytosine and thiamine. The addition of the sodium chloride was also done to the hydrazine reaction. Hydrazine reaction you are familiar with for cytosine and thiamine. Why the Maxam-Gilbert added the sodium chloride? The reason is that to inhibit the reaction of the thiamine, and for the C only reaction, means to inhibit for the thiamine just for the C, for the cytosine. The reaction will be for the cytosine and inhibited by the thiamine. So it was also added in some reactions by the Maxam-Gilbert. So the basic concept about that is the formic acid and dimethyl sulfate is used for the purine bases, and the hydrazine used for the pyrimidine. I am repeating again and again. So the concept clear upon you people? Okay, now I am going to the next step. Now what will happen after the chemical? Now the whole fragment is chemically modified at different nucleotide or at different nitrogenous bases, mean adenine, guanine, cytosine and thiamine. Now what will happen? The modified DNA may then be cleaved by the piperidine. Now what will happen? That modified DNA is cleaved by the piperidine at the position of the modified base. Now I am showing you here. This is the DNA, single stranded DNA. You can see here at the 5 end there is a radioactive isotope, and now what will happen? The Maxam-Gilbert modified the DNA at G position, at C position, and at the T positions, and also with A plus G, means in one tube modification was done at the A and the G position, mean adenine and guanine, and in the second tube the modification was done at the G position. Like dideoxy. In case of the Sanger method, the Sanger used the dideoxy to stop or to terminate the DNA elongation. Here what will happen? The chemical modification occur at different position along the DNA strand. Now you can see here the first A plus G means modification will be occur at the adenine and guanine, and just in this case in the second reaction you can see here the modification occur only at the G position, and in the C reaction what will happen, the modification occur with, at the C position, and in the C plus T reaction the modification will be occurred or was occurred at what, C by the Maxam-Gilbert at C position and the T position. Now CT, AG, you are familiar, the adenine, guanine, cytosine, thymine. So the DNA is made up of all these nitrogenous bases are the nucleotides, and the modification occur at these nucleotides along the DNA molecule by different chemicals like the formic acid, dimethyl sulfate, and hydrazine. Now what will happen? When the modification will occur here along the DNA strand, then the next step will be the piperidine application. And piperidine is used for what? For cleavage of the DNA. Now if the DNA is modified at the G position, then it will break the DNA at this position. DNA strand at this. If the modification occur at the G position, A position, then it will break the DNA molecule at the A position. Similarly with the C, if the modification occur at the C, if similarly for the T, where there is a modification by the chemical, the piperidine cleave that DNA molecule at that position. Now I hope so the concept about the chemical modification and the cleavage will be clear upon you people. Is it clear now? That is very interesting. Okay. Now what will happen after the cleavage and using different, now I'm showing you. Now what will happen after modification and the cleavage by the piperidine? What will happen? Different fragments will be produced. Now you can see here with A and G, when the modification occur at the G position, now no fragment will be produced, because at G the modification if occur at the G, then there is no strand formation, or you can say that the nucleotide attachment occur here. So stopped here. So at G there is no fragment will be produced. So you can see here A plus G. Now I am going to give you the concept of A plus G. What will happen? First of all, I am giving you the concept of the G position. So when the modification occur at the G, then there is no fragment will be produced. Okay. Now you can count the G. G is here. G is here. Is it clear? So the first fragment must be produced here, but not produced here, because this is the single. So at G, the after modification, what will happen? The DNA formation will be stopped here. Now what will happen? The second step, the modification occur at this G. Now you can see here the DNA strands, other strand will be produced here, G plus C plus T plus A plus C. So the modification occur at that G. So the fragment will be produced here, G, C, T, A, C. Is it clear? So first here we will stop here. No fragment will be produced at this G, but at this G the fragment will be produced with G, C, T, A, C. So the break will occur by the piperidine here. 1, 2, 3, 4, 5. 5 basis fragment will be produced. Now with the A. Now what will happen with the A? Now count the A. A is here. A is here. So the first modification occur here. And the fragment will be GCT. You can see here the fragment GCT is produced. And the next fragment will be A is here. So you can count GCT, ACA, CGT. So you can see here. First fragment, second fragment, and third fragment. Now the consider. Now for the G, just for the G, 2G are here. At this G, there is no fragment is produced, but at this G, there is a fragment is produced which contain the GCTAC. GCTAC. Now for the C, you can see here C is here. So it will stop here and just one fragment will be produced containing only G. Is it clear? Now where is the C? Here is the C. You can count GCTA. GCTA. Second fragment in the C tube will be with GCTA fragment, and G fragment in the G just GCTAC, and in the A plus G GCT, GCTAC, 3 fragments are produced. Now in the fourth tube you can see C plus T. So C first modification occur here, so only just one G fragment is produced here, and then with the C you can see here C with here C. So GCTA, and the third one C is okay. No, now we are counting for the T. T is here. T is here. So the first fragment GC. The second fragment T is here. So the GCTACG. You can see here. Is it clear? So after that, all these 4 reactions are now what? They are loaded on the gel. And the gel was labeled by the Maxam-Gilbert as A plus G lane. This second lane is G, C, C plus T. So 4 reaction, 1, 2, 3, 4 reaction, 4 tubes mixture is loaded. First one is here, second one is here, third one is here. And just like the second method, we will do the electrophoresis and autoradiography similar to that. So I will not discuss here again. You must read from this slide. Okay. Now the reading of the sequence. The reading of the sequence is same like the second method. We start from the small fragment. Now you can see here which fragment is small. This G fragment is small, this G fragment is small. Is it clear? With the C. So we label the gel with C, T, A, C, G, T, A. You can see here we can easily read the sequence from the gel. When we put we load the 4 tubes on the gel, the DNA fragments move and the smaller fragments will move away from it, and so first fragments we will count is the smallest fragment, then the second smallest fragment, then the third, then the fourth, then the fifth, then the sixth, and then the seventh. So you can read all these sequences in this way. Is it clear? Now you can easily know the C, T, A, C, G, T, A, and you can also read from this figure here which fragment is the smaller one. G is the smaller one fragment, then the next fragment GC, you can see here in the CT position, CT at tube reaction, and the third one is the where GCT3, 1, 2 base pair is here, 3 base pair is here, 4 base pair is here, like that. Very simple. Is it clear to all of you? Okay. Now you can read from the slide. To read the sequence, you begin with the smaller fragments at the bottom of the gel, calling each base involved interpreting the band pattern relative to the 4 chemical reaction. Is it clear now? For example, if a band in the DNA sequence appears in both the G reaction and the G plus A reaction, then that nucleotide is a G. G and G plus A, you consider it as a G nucleotide. If a band in the DNA sequence appear only in the G plus A reaction land, then it is an A. As you can see here, yes, you can see here, this is the G, means the C, because the modification occur at the C. This is for the C, this is for the T, this is for so on. Okay. The same decision process work for the C reaction and the C plus T reaction land. Sequences are confirmed by running replicate reaction on the same gel and comparing the autoradiogram pattern between the replicates. So this is the overall Maxam-Gilbert method. I hope so the lecture will be clear upon you people. If you have still any confusion, any question, you can ask me. I will be back with the new lecture. Thank you very much. Till then, Allah Hafiz.